Welcome back to the deep dive. I have to admit, the research we are covering today kept me up last night.
Oh, yeah.
Yeah. Not because it's scary, though. Honestly, the implications are a little vertigo-inducing, but because it feels like we just found a glitch in the matrix.
I know exactly what you mean.
We are looking at a convergence between two completely different worlds that, by all rights, shouldn't really have anything to do with each other.
It is one of those rare stories that makes you pause and question the nature of invention. You start asking yourself, how much of engineering is us actually creating something new, and how much is just us finally stumbling upon a blueprint that's been sitting there the whole time?
Exactly. It's the difference between invention and discovery.
Yeah.
And today, the line between those two is going to get very, very blurry.
It's going to get erased, I think.
So let's set the stage for everyone listening. We need to go back about a year to January 2025.
January 2025, a massive month for physics.
That is when a company called PsiQuantum published a paper in Nature. Now, we cover a lot of tech breakthroughs on this show, and usually it's some incremental step. We made the processor 10% faster.
Right. A little tweak here, a small efficiency gain there.
But this was different. This paper basically shook the foundations of the computing world.
It was a watershed moment. For decades, quantum computing has been this always 10 years away technology. It's been the domain of physicists in university basements dealing with, absolute zero temperatures and magnetic traps.
The stuff of science fiction, really.
Exactly. But PsiQuantum dropped this paper describing a manufactured chipset for a fault-tolerant photonic quantum computer. And the key word there, the one that changes everything, is manufactured.
Right. We aren't talking about a delicate experiment held together with duct tape and hope.
Not at all.
They made this chip at global foundries. For people who don't follow the semiconductor industry, that is a massive commercial foundry. It's the same kind of facility that churns out the chips for your phone or your laptop.
That is the big leap. They moved from the lab bench to the assembly line. They showed that you can build the most complex machine in human history using the same supply chain that builds a PlayStation.
That's a powerful way to put it.
But, and this is the hook for our deep dive today, underneath that manufacturing triumph, there was a hidden physics problem they had to solve. A problem that is fundamental to using light for computation.
And this is where the story gets wild. Because the solution they came up with, the specific architecture they built to make this computer work, turns out to be the exact same architecture that has been running inside our own bodies for 2 billion years.
It is the parallel architecture. And when we say exact same, we don't mean it as a vague metaphor. We aren't saying, oh, the computer has a brain and you have a brain.
No, this is much more specific.
We mean the structural logic, the engineering steps are identical, point for point.
So on one side, we have the Omega chipset, the most futuristic machine humans have ever built.
The absolute frontier of technology.
On the other side, we have the mitochondria inside our cells.
These ancient biological engines.
And they are doing the same thing.
To solve the exact same problem.
Okay, let's unpack this. We have to start with the machine. What exactly did PsiQuantum build, and why was it so hard? Why couldn't they just build a normal computer, but with quantum parts?
So PsiQuantum is building a photonic quantum computer. Most people, when they read about quantum computing, they hear about superconducting qubits, like what Google or IBM use.
Right, those giant chandelier-looking things.
That's them. Those are little circuits made of metal, niobium usually, chilled down to absolute zero in these giant dilution refrigerators. They sit there static, and you manipulate them with microwaves.
But PsiQuantum uses photons, particles of light.
Correct. And photons have some massive advantages over the metal circuits. First off, they don't decohere easily.
Explain decoherence for us. It's a word we hear a lot, but I'm not sure everyone gets what it means.
Decoherence is the enemy of quantum computing. It's when the environment interacts with your qubit, a little bit of heat, a stray magnetic field, a vibration, and causes it to lose its quantum state.
It stops being quantum.
It turns from a magic quantum coin that can be heads and tails at the same time into just a regular old penny. It's either heads or tails, game over.
And superconducting qubits are very sensitive to that.
Incredibly sensitive. But photons are antisocial. They don't interact with much. You can shoot a photon through miles of fiber optic cable, and it keeps its state perfectly.
Plus, they travel at the speed of light.
Exactly. And as we mentioned, you can print the waveguides, the little tracks that guide the light using standard silicon manufacturing. No exotic materials needed.
So that all sounds great. It sounds like photons are the obvious choice.
Right.
Where's the problem?
But there is a catch, a huge catch.
There's always a catch.
The catch comes down to generation. With a superconducting qubit, the qubit is a physical object. It is a loop of metal sitting on the chip. You can point to it, it's always there.
It's a permanent fixture.
Right. With a photon, you have to create it. You have to generate your qubit fresh every single time you want to use it.
And you can't just press a button and say, give me one photon.
You cannot. This is the generation bottleneck. And to understand it, we have to look at the physics they use. It's a process called spontaneous four-wave mixing, or SFWM.
Okay, stop right there. Four-wave mixing. That sounds like a DJ technique. Break that down for us. Why is it called four-wave?
It's actually pretty straightforward math, rooted in the conservation of energy. Here is the setup. You have a silicon ring resonator, a tiny loop etched on the chip, smaller than a human hair.
Okay.
You pump a powerful laser into it. That laser light is made of photons, of course. In this process, you take 2 photons from that strong pump laser. That's 2 waves.
2 waves in.
You smash them into the crystal lattice of the silicon. And through the nonlinear optical properties of the silicon, those 2 pump photons disappear. They are annihilated.
Gone.
And their energy is re-emitted as 2 new photons at different frequencies. One is slightly higher energy, one is slightly lower.
So 2 photons go in, 2 new photons come out. That's 4 waves total.
Exactly. 2 ins, 2 out. 4-wave mixing. One of the new photons is called the signal. That's the one we want to use for our computer. The other is called the idler.
Signal and idler. Got it. Okay, so we have a mechanism. We pump laser light in. We get photon pairs out. Why is this a problem?
Because of the word spontaneous, this process is probabilistic. It is inherently random.
What does that mean in practice?
Think of it like a coin flip, or more accurately, a weighted die. You pulse the laser, and most of the time nothing happens. The pump photons just pass right through.
Nothing.
Then sometimes you get a pair. But you can't predict when.
So you can't guarantee it will happen on any given pulse.
No. And here's the kicker. You might be thinking, well, just turn up the power, blast it with more laser until you're guaranteed a pair.
That seems like the logical fix, yeah. Just overwhelm it.
But you can't. If you pump it too hard, you get multi-pair emissions.
No meaning.
You get 2 pairs or 3 pairs at the same time. And in quantum computing, that is noise. It's garbage. It ruins the calculation. You need exactly 1 pair, a single clean event.
So you have to keep the power low to avoid noise.
Exactly. You have to keep the probability low.
Which means the probability of getting a pair is low.
Extremely low. You have to operate in a regime where most of the time you fail.
So this is the fundamental flaw.
Yeah.
You are trying to build a computer which needs to be the most reliable thing in the universe. 1 + 1 must equal 2 every single time.
Every time.
But your fundamental building blocks, the photons, are flaky. They mostly fail to show up when you ask for them.
Precisely. Imagine trying to build a brick wall, but every time you reach for a brick, there's only a 10% chance one is actually in your hand. How do you build a solid wall?
You can't. Or, I guess you'd have to try thousands of times just to lay one course.
And a quantum computer needs millions of these bricks per second, all arriving on cue.
That is the central question of this deep dive. How do you build a perfectly reliable machine from unreliable parts?
And PsiQuantum solved this with a specific architectural strategy. And this is where we need to pay close attention, because this is the blueprint we are going to see again later. It's a 3-layer solution.
Okay, walk us through the layers. Layer 1.
Layer 1 is heralding or verification. Remember I said SFWM creates a pair of photons. The signal and the idler.
Right. The signal is the qubit we want. The idler's the byproduct.
Well, PsiQuantum turns the byproduct into a tool. The 2 photons are created together. They're twins, so they get split onto 2 different paths on the chip.
Okay.
You send the signal photon into a storage loop, basically a little racetrack, a delay line, and you send the idler photon straight to a detector.
You're checking the receipt.
Precisely. That's the perfect analogy. If the detector clicks, it is a herald. It tells you, hey, the idler is here. Because they're twins, you know with near certainty that its partner, the signal photon, exists and is sitting in that waiting room, that racetrack.
And if it doesn't click...
If the detector doesn't click, you know that attempt failed. You didn't get a pair. So you just discard that time slot, you flush the system and try again.
So it turns an unknown into a known. You aren't guessing if you have a qubit anymore. You have verified it.
You've turned a probability into a certainty. And PsiQuantum's detectors are incredible, by the way. These are superconducting nanowire single photon detectors. They achieve about 98.9% efficiency. So if that idler is there, they will see it.
That's amazing. But, okay, so now we know which attempts worked. But that doesn't solve the problem that most of them don't work, right?
Okay.
If I need a photon right now to do a calculation and my generator fails, even if I know it failed, I still don't have a photon. The computer crashes.
That brings us to layer 2, multiplexing, or simply put, massive parallelism.
Yeah.
Since you can't rely on one source, you build thousands of them.
The strategy of numbers. Brute force.
Think of it like a lottery. If you buy one ticket, your odds are terrible. If you buy every ticket in the store, you are guaranteed a win. PsiQuantum doesn't rely on one ring resonator. They have thousands of these sources on the chip, all pulsing at the same time.
All of them firing away, trying to generate a pair.
Right. Statistically, even if each individual source has a low success rate, if you run enough of them in parallel, you can guarantee that at least one of them will produce a heralded photon in any given time slice.
So you've guaranteed a winner somewhere on the chip.
Yes.
But it might be on the left side of the chip, and you need it on the right side for the next step of the calculation. How do you get it there?
That is the second half of multiplexing, the switch. PsiQuantum built these active optical switching networks. Think of it like a massive train yard.
Okay.
The photon is the train. We don't know which track it's coming on. Remember, it's probabilistic. But the moment it arrives, the moment we get that herald click from the detector, the switch tracks slam into position to route that specific photon to the main line to where the computation is happening.
And this has to happen fast.
Nanoseconds. Before the photon finishes its loop in the delay line, the path is already set. So you are converting a probabilistic resource, something that might happen, into a deterministic supply. Something that is guaranteed to be there when you need it.
By using thousands of sources and a smart, ultra-fast switching network.
That is the core of the architecture. It's genius. But there is a layer 3, and this is specific to how they compute. It is called fusion-based computing.
This sounds like sci-fi, fusion-based computing.
It does, but the idea is actually quite elegant. In this model, photons aren't like the bits in your laptop that sit there holding a 0 or a 1.
They're not static.
No, they are consumed. You take these heralded photons you've just routed, you fuse them together using what we call destructive measurements.
Destructive. That sounds bad, like you're breaking it.
It sounds bad, but it's essential. In quantum mechanics, measuring something usually collapses its state, right? It destroys the quantumness.
Right, the observer effect.
Here, they use that collapse to their advantage. They entangle the photons, measure them in a very specific way, and the pattern of those measurement outcomes is the computation. The photons are fuel. They are used up to create the logic.
So the machine is a furnace. It is constantly generating photons, verifying them, routing them, and then burning them up to process information.
That is the PsiQuantum machine. It's a continuous flow process. And the numbers from that January 2025 paper are just staggering. This is all strictly Tier 1 established science. They achieved 99.98% fidelity in propelling these states.
That's incredibly pure.
And they have a loss tolerance, meaning they can lose about 10.4% of the photons during the fusion process and still get the right answer. It is a marvel of modern engineering built to survive in a noisy world.
So we have the blueprint. Let's recap it because this is crucial for what comes next.
Yes, let's lock it in.
1. Probabilistic generation. You create raw materials, in this case photons, via a random process, SFWM.
Check.
2. Verification or heralding. You check a receipt, the idler photon, to confirm the material exists and is good to go.
Check.
3. Multiplexing. You run thousands of generators in parallel and use switches to route the winners, turning a maybe into a certainty.
A deterministic supply from a probabilistic source. Check.
And 4. Consumption. You burn the resource in a destructive measurement to do the work.
That's the architecture.
Right. Keep that 4-step list in your head. Probabilistic generation, verification, multiplexing, consumption.
Now, I want you to close your eyes. Well, don't close your eyes if you're driving. But imagine we are shrinking down. We are leaving the clean room at Global Foundries. We are leaving the silicon and the lasers. We are going inside your own body.
Into the cell.
Into the cell, and specifically into the mitochondrion.
The powerhouse of the cell. I think that's the only thing everyone remembers from high school biology.
It's a cliche, but it's true. But we are going to look at it not as a biologist would, but as an engineer would. We are looking at the inner mitochondrial membrane. This is where your body generates the energy to keep you alive.
And this is where we find the parallel.
This is where we find Act 2, the unlikely source. The primary mechanism for generating energy in your body is the electron transport chain. Basically, you strip electrons from the food you eat and you pass them down a chain of proteins embedded in this membrane.
Like a bucket brigade.
Exactly like a bucket brigade. But here's the thing that most textbooks gloss over. These proteins are separated by gaps. Physical gaps of about 14 angstroms.
How big is that, really?
It's tiny, about 1.4 nanometers, but for an electron trying to move via classical physics, it might as well be the Grand Canyon. It is too far to jump. It can't happen.
So how do they get across? If they can't jump, do they flow?
They tunnel. Quantum tunneling.
Okay, hold on. We hear quantum used in biology a lot these days, and sometimes it feels like hype. Like people putting the word quantum in front of consciousness to make it sound magical.
And you're right to be skeptical.
Is this real quantum mechanics? Or a metaphor?
I am glad you asked that because skepticism is healthy. But this is tier 1, solid, peer-reviewed physics. This is not a metaphor. This was established by Christopher Page, Christopher Moser, and Leslie Dutton back in 1999. They published the paper in Nature describing what we now call the Moser-Dutton ruler.
The ruler.
It is a graph. It plots the rate of electron transfer against the distance between the molecules, and it shows, definitively, that electron transfer rates in biology drop off exponentially with distance. It's a perfect match for the equations of quantum tunneling.
So biology uses the same math.
It's not just that it uses the math, it's that nature has tuned the distance between these proteins, specifically the little iron-sulfur clusters inside them, the redox cofactors, to be exactly within the range where quantum tunneling is efficient.
So it's not random, it's engineered.
It is precise engineering. If the proteins were a little further apart, the electrons wouldn't tunnel fast enough and you would die of suffocation essentially instantly. If they were closer, you'd get short circuits, the electrons would leak. Nature hit the sweet spot around 14 angstroms.
So my metabolism, the very act of being alive, relies on particles teleporting through barriers they shouldn't be able to cross.
It is not a metaphor. It is genuine quantum mechanical tunneling. In fact, in cytochrome cyoxidase, that's the final enzyme in the chain, the one that actually uses the oxygen you breathe, the electron tunneling happens on the nanosecond scale. It is incredibly fast.
Wow.
And it gets even wilder. It's not just electrons. Protons tunnel too. There was a study that showed a kinetic isotope effect of 7.7 for proton transfer in that same enzyme, which is, well, it's smoking gun proof of proton tunneling.
Okay, but wait. Let's go back to the first step of our architecture.
Yeah.
Just like the photons in the quantum computer, isn't tunneling probabilistic?
It is. That is the fundamental nature of quantum mechanics. Tunneling is a stochastic process. You cannot predict exactly when a single electron will tunnel from one protein to the next. It is random.
So we have the same starting problem. We have a probabilistic generator.
Yeah.
In PsiQuantum, it was a random photon pair. Here, it's a random electron tunnel.
Exactly. And just like PsiQuantum, biology needs a reliable output. You can't have your heart stop beating just because the electrons decided not to tunnel this millisecond. The system needs to be deterministic.
So what is the biological equivalent of layer 1? The verification. In PsiQuantum, it was the idler photon clicking a detector.
In the mitochondrion, verification is the proton gradient.
Explain that. How is a gradient a verification? Gradient is just a difference in concentration, right?
Right. But think about what the electron transport chain actually does. It doesn't just pass the electron for fun. Every time an electron successfully tunnels through the chain and completes a catalytic cycle at one of the protein complexes,
Yes.
the protein uses that energy to pump a proton across the membrane, from the inside to the outside.
It moves a charged particle from one side to the other.
Right, it charges the battery. But think of it in terms of information. If the proton is successfully pumped, it is a physical verification that the quantum event, the tunneling, occurred and was successful. It is a click of the detector.
Ah, I see. The successful quantum event has a direct physical consequence.
Yes. And that consequence, the pumped proton, contributes to this massive shared buffer. It integrates millions of these random probabilistic quantum events into a smooth, reliable potential.
So the jagged, noisy, random tunneling events get smoothed out into a steady voltage, a steady pressure.
That is the multiplexing, layer 2. A single mitochondrion doesn't rely on one electron transport chain. It has thousands of these chains running in parallel, all embedded in its inner membrane.
And a single cell.
A single cell has hundreds or thousands of mitochondria, and recent work shows cells actively manage these populations, specializing some for high output, others for different tasks. It's a whole power grid.
There's the massive parallelism.
You are running billions of probabilistic generators simultaneously. The proton gradient is the swishing network. It takes the energy from whichever chain happened to fire successfully and stores it in a common pool that the cell can use.
It effectively converts a probabilistic resource into a deterministic supply.
You were saying the exact same sentence we used for the quantum computer.
That is eerie. It really is. Okay, so we're building the parallel.
Let's lay it out.
We have generation, electron tunneling. We have verification, proton pumping. We have multiplexing the gradient and the whole network of mitochondria. Now we need layer 3, consumption, deployment.
Right.
In the computer, this is the fusion gate, consuming the photon to do work. What is it in the body?
It is ATP synthase.
I remember the name, but I feel like we're about to go deeper than the textbook diagram.
ATP synthase is my favorite protein in all of biology. I think it's the most beautiful piece of machinery on the planet.
Why? What does it do?
It's the machine that uses that proton gradient, that pressure you've built up to make ATP. And when I say machine, I mean that literally. It is a rotary motor.
A motor, like spinning.
Physically spinning. It has a rotor called FO embedded in the membrane. Protons flow through it from the high concentration side to the low concentration side, and the force of that flow, the proton motive force, causes it to spin.
Like water tuning a turbine at a dam.
It's a perfect analogy. And that rotor is connected to a shaft, the gamma subunit, which extends up and spins inside a stationary head called F1.
How do we know it spins? Can we see something that small?
We can now, but the definitive proof came in 1997. It's one of my favorite experiments ever. A team led by Hiroyuki Noji did something incredible. They isolated the motor, stuck it to a glass slide upside down, and attached a tiny fluorescent filament of actin to the spinning shaft.
Like putting a piece of tape on a fan blade so you can see it moving.
Exactly. And then they put it under a microscope and fed it ATP to make it run in reverse, and they watched it. They literally video recorded it spinning counterclockwise.
That is incredible. A microscopic motor inside us. That's not a metaphor. They watched it happen.
And the stats, this is where I get excited. Let these numbers breathe for a second. This motor spins at about 130 revolutions per second.
130 times a second.
That is roughly 8,000 RPM.
8,000.
8,000 RPM. That is comparable to a Formula One engine redlining.
But it's what, a few atoms wide?
It is doing this at the nanometer scale. It is only about 10 nanometers across.
And efficiency. A Formula One engine wastes an enormous amount of heat.
A car engine is maybe 20% or 25% efficient. A really good electric motor is great, maybe 95% efficient.
Right.
ATP synthase operates at nearly 100% thermodynamic efficiency. The energy in equals the work out. Friction is almost non-existent at that scale. It is a physically perfect machine.
That is tier 1 science, all of this.
All tier 1 established science. A later paper from Yasuda and colleagues in 2001 measured the steps. They saw it rotate in discrete 80-degree and 40-degree substeps, perfectly timed with the chemistry it's performing.
So as it spins, this motor is, what, smashing molecules together?
Yes. The spinning shaft bumps into the parts of the stationary head, causing them to change shape. And these shape changes force a molecule of ADP and a phosphate ion together to create one molecule of ATP. It's mechanical synthesis.
Click, click. 1 ATP per step.
3 ATP per full rotation.
So the spinning motor is creating ATP. We know ATP is energy currency.
Yeah.
But how much of it are we talking about?
This is the stat that usually breaks people's brains.
I'm ready.
You, sitting there right now, produce roughly your own body weight in ATP every single day.
Wait. No, they can't be right. I weigh host weight. I don't make host weight of anything in a day.
You do, every day. Now, you don't store that much. You'd explode. You only have about 50 grams of ATP in your body at any one time.
Just a few ounces.
Right. That means you are recycling that entire pool 500 to 700 times a day.
That is relentless, a constant churn.
It is continuous, massive-scale manufacturing. And every single molecule of that ATP, every gram of that body weight, traces its origin back to a quantum tunneling event in the electron transport chain.
So let's look at the full parallel again. This is the aha moment.
Let's do it.
PsiQuantum uses probabilistic photons generated by SFWM. Biology uses probabilistic electrons that quantum tunnel.
Point 1, matched.
PsiQuantum uses heralding detecting an idler photon to verify. Biology uses proton pumping, the successful completion of a catalytic cycle, to verify.
Point 2, matched. And we should be honest here, the mechanisms are different. Heralding is a single quantum measurement. Proton pumping is the end result of a classical cycle driven by quantum events. But the functional role...
The functional role is identical. It's a verification step.
Exactly.
PsiQuantum uses thousands of sources and optical switches to multiplex. Biology uses thousands of electron transport chains, hundreds of mitochondria, and the proton gradient to multiplex.
Point 3, matched.
So quantum consumes photons in fusion gates to do work. Biology consumes the proton gradient in ATP synthase to do work, creating ATP.
Point 4, matched.
And there's one more nuance to the parallel: context-dependent deployment.
Yes, this is crucial.
In the quantum computer, the photon is a standardized resource. It doesn't know what calculation it is part of. It just gets routed to a fusion gate to become part of an AND gate or a CNOT gate.
Right, it's a generic qubit.
In the body, ATP is a standardized resource. It is the universal energy currency. The ATP molecule made in your heart doesn't know it's in the heart. It just gets routed to a myosin protein to make a muscle contract.
Or if it's in the brain, it gets routed to an ion pump to help you think. Or to a helicase enzyme to unwind DNA.
The work packet is identical, but the context of its deployment changes completely.
Exactly. The architecture is build a reliable system from unreliable parts via parallelism and standardization.
Okay, I want to clarify something for the listener, though. Are we saying PsiQuantum copied the mitochondria? Is this biomimicry?
No, that's the most fascinating part of the whole story. This is independent discovery.
They didn't know.
The PsiQuantum engineers didn't look at a biology textbook. They weren't trying to copy a cell. They were trying to solve a hard physics problem. They pushed the physics of light to its absolute limit. And when you push physics to the limit, the solution space narrows.
There are only so many ways to solve the problem.
Exactly. In some cases, there is often only one optimal way to do it. And it turns out evolution found that optimal way 2 billion years ago.
So we have established that this is happening.
Yeah.
The engineering blueprint matches the biological blueprint. The question is, why? Why does this keep happening?
This is where we shift gears, Act 3. And I want to be very clear with our listeners here.
Yeah.
Everything we have discussed so far, the tunneling, the spinning motor, the photon heralding, the stats, that is all Tier 1 established science. Factual observation. Now we are moving into the why. And this ranges from well-supported theory to open speculation.
Okay, we are entering the speculation zone. Let's start with the most solid explanation, the Tier 1, Tier 2 layer. Engineering constraints.
Right. This is Ashby's law of requisite variety. It's a principle from cybernetics published back in 1956 by a brilliant thinker named W. Ross Ashby.
What does the law say?
It says simply, only variety can destroy variety.
Okay, that's a bit of a Zen-kane.
It is. In engineering terms, it means a controller must have at least as many states or as much flexibility as the disturbances in its environment.
That sounds abstract. Give me a real-world example.
Think about a simple thermostat in your house. It has 2 states, on or off. If the temperature outside fluctuates wildly, sun, clouds, snow, wind, that simple on-off controller struggles to keep the temperature inside perfect. It's always overshooting or undershooting. It doesn't have enough variety to match the environment's variety.
Right.
Now think about the environment inside a cell. It is chaotic. Oxygen levels fluctuate. Nutrient availability spikes and crashes. Demand for energy skyrockets when you stand up. There's noise everywhere.
And the quantum computer.
The same. Thermal noise, photon loss in the waveguides, imperfect detectors, electromagnetic interference. Both systems are operating in high noise, high disturbance environments. They have a huge amount of variety to deal with.
So a single linear system would fail.
A single generator like one photon source or one electron chain would get overwhelmed. It doesn't have the requisite variety to handle the noise. But if you have thousands of generators running in parallel, you have massive variety.
Your controller now has thousands of states.
Exactly. You can absorb the fluctuations. You can lose 10% of your photons or have 20% of your electron chains stall, and the system keeps running smoothly. Ashby's law suggests that this architecture, massive parallelism, is one of the only ways to build a reliable system in a chaotic environment.
So they didn't just happen to look the same. The laws of control theory forced them to look the same.
That is the Tier 1, Tier 2 explanation. The constraint forces the convergence. The problem is so hard, there's only one shape of solution.
Okay, what is the next layer of explanation?
The next layer is thermodynamics, specifically the thermodynamics of information. This is Landauer's principle.
We have touched on this in previous deep dives. Information isn't free, there's a physical cost to it.
Exactly. Rolf Landauer proved in 1961 that erasing information, or in this case, making a measurement to gain information, has a minimum energy cost. There is a heat tax you have to pay equal to kTln2. It's a fundamental law of physics.
And this has been experimentally verified, right?
Oh, yes. A group in 2012 did a beautiful experiment with a single silica bead in an optical trap and measured the heat released when they erased 1 bit of information. Landauer was right.
So how does this apply here?
In the quantum's machine, every time a detector clicks to herald a photon, they are gaining information. They're going from maybe there's a photon to yes, there is a photon. That information gain costs energy. They have to cool that superconducting detector, which costs a ton of energy.
They're paying the Landauer tax.
Yes. And in the mitochondrion, every time a proton is pumped to verify the tunneling event, it costs energy. In fact, that proton is the energy storage. The system is paying a tax to turn the uncertainty of a quantum tunnel into the certainty of a pumped proton.
So both systems are paying an energy tax to turn uncertainty into certainty.
And Landauer's principle sets the floor for that tax. It tells us that this problem, getting reliable work from unreliable parts, is fundamentally an information processing task. It is not just about energy, it is about knowledge. And because it is an information task, it is bound by the fundamental laws of thermodynamics.
So Ashby dictates the structure, and Landauer dictates the cost.
That's the way I see it. Those two principles corner you into this solution.
Now let's get into the Tier 3 stuff, the deep speculation. You mentioned earlier something about a clock signal.
This is a fascinating open question. This is where we're really on the frontier. We know cytochrome C oxidase, that final enzyme, turns over about 1,000 times a second. That's a 1 kilohertz cycle, a millisecond timescale.
Okay.
Well, in the brain, a lot of important neural processes, like the firing rates of neurons and synaptic plasticity, also operate on millisecond timescales.
Same frequency.
The same frequency range. So some researchers, and this is conjecture, have asked, is the mitochondrial turnover rate just a kinetic speed limit? Or is it a clock?
Like the clock speed in a CPU, a quartz crystal ticking away at a set frequency.
Exactly. Is the metabolic machinery in our neurons actually providing a timing signal, a beat that helps to synchronize neural firing?
So the power supply is also the metronome.
It's a tantalizing idea. We don't know. It is a completely open question. But if it were true, it would mean the convergence goes even deeper, that biology isn't just generating power, it is generating the beat for the computation of the brain.
That is wild to think about, that our thoughts might be synchronized to the spin of these molecular motors deep inside ourselves.
It is a possibility. But again, Tier 3, we do not have the smoking gun for that yet. Not even close.
So there's one more big question here, a really deep one. Is there a missing theorem?
This is what keeps me up at night. This is the big what if. We see this convergence. We have Ashby and Landauer pointing us in the right direction. But is there a formal mathematical theorem, one we haven't discovered and proven yet, that mandates this architecture?
A theorem that says any system that reliably extracts classical work from probabilistic quantum events must use a massively parallel architecture with verification and routing.
Exactly. If such a theorem exists, it would explain why PsiQuantum and mitochondria look identical. It wouldn't be a coincidence. It wouldn't just be a good engineering idea. It would be a law of the universe.
It would be like the laws of thermodynamics. You
Right.
can't build a perpetual motion machine.
And maybe you can't build a quantum classical bridge any other way. Maybe this is the only stable, scalable solution.
It is the one best way theory. And we see this elsewhere in nature and engineering, don't we?
All the time. Look at flight. Birds, bats, insects, and airplanes. They all use wings. Not because they copy each other, but because the physics of air, the Navier-Stokes equations, demand wings for efficient lift. Look at eyes. The camera eye evolved independently in vertebrates like us and in cephalopods like octopuses. They're on completely different branches of the evolutionary tree, yet their eyes are almost identical in structure. Why? Because the physics of light demands a lens, an iris, and a retina to form a high-resolution image.
And now we are seeing it at the most fundamental level in quantum engineering.
It is independent discovery. When you are constrained by the same laws of physics, you often arrive at the same solution. See, quantum pushed the physics of light to its limit. Evolution pushed the physics of electron transfer to its limit, and they met in the same place.
It's humbling. We think we are inventing the future, but we are really just uncovering the past.
Or uncovering something timeless. It's data. It proves that biology isn't magic. It is engineering. Just engineering that has had a 2 billion year head start on us.
So let's recap the confidence tiers before we wrap up, just so everyone is clear on what is fact and what is theory.
Sure.
Tier 1, the solid ground.
Tier 1, solid fact. Electron tunneling in the electron transport chain. The Moser-Dutton ruler confirming it, ATP synthase as a rotary motor spinning at 8,000 RPM, the PsiQuantum architecture heralding, multiplexing, fusion, and the structural 4-part match between them. That is all real, published, peer-reviewed science.
Okay. Tier 2, the likely explanation.
Tier 2, likely. Ashby's law of requisite variety and Landauer's principle as the reason for the match. This is the best explanation we have, and it is well supported by fundamental physics and control theory, but we haven't formally proven it in this specific biological context. It's a very strong inference.
And tier 3, the frontier.
Tier 3, unknown. The idea of a biological clock signal derived from metabolic rates or a universal quantum to classical theorem, these are the frontiers. These are the exciting open questions for the next generation of scientists to figure out.
I love that. We have the answer to what and a pretty good handle on why, but the deepest why is still up for grabs.
That is the beauty of science, isn't it? There is always another layer to peel back.
So, final thoughts. We built a machine to process quantum information using light. It is arguably the most advanced technology on Earth. And it turns out we are walking around with trillions of versions of that machine's core architecture inside us right now.
It really reframes what it means to be human, I think. It stops being us versus nature or technology versus biology. We are tech. We are machines built in the same physical principles as the things we create. And the fact that we converged on the same design isn't mystical. It's validation. It means we are on the right track. It means our understanding of physics is getting so deep that we are arriving at the same conclusions as a 2-billion-year optimization process. If we build it completely differently than biology, I'd be worried we got it wrong.
That's a great point. The convergence is the proof.
Exactly.
Here is a question to leave you with, listener. If the quantum computer architecture was already solved by biology, what other inventions are we struggling to build right now that are already running inside of us? What is the next convergence waiting to be discovered?
That is the billion-dollar question. Or trillion-dollar, maybe.
Thanks for diving deep with us.
Always a pleasure.
See you in the next deep dive.